Hello everyone, my name is Gabriel Kupovics. I'm a student at the University of Arkansas in Fayetteville, Arkansas. And today I'll be presenting some research that's being performed in Dr. Suresh Kumar's lab with the help of Zeina Alraawi. So today we'll be talking about the effects of self-assembled structures of phenylalanine on the amyloid-like fibril formation of human acidic fibroblast growth factor, or hFGF1. So before I get into the project, I would like to say that I realize that this is probably pretty difficult to see in the video, but there should be a link below to a PDF file so that you can follow along. So the importance of this research begins with a discussion of phenylketonuria, also known as PKU. This is an inborn error of metabolism that results from a mutation in the PAH gene, which codes for phenylalanine hydroxylase. This enzyme is responsible for beginning the metabolism of phenylalanine, and when it's absent or mutated, phenylalanine is allowed to accumulate in the body, leading to nerve cell sensitivity. This sensitivity can lead to cognitive disability in the individuals. Unfortunately, the etiology and mechanism of PKU is not entirely known, so this research begins to explore that. Additionally, Alzheimer's disease, which is probably more well known, the etiology is largely debated, so we'll be talking a little bit about Alzheimer's as well. So hFGF1 is a prototypical member of the FGF superfamily of cell signaling molecules. These are responsible for wound healing, angiogenesis, and importantly for this research, neurogenesis. Interestingly, studies have found elevated levels of hFGF1 in the cerebrospinal fluid of Alzheimer's patients. This leads us to believe that FGF1 might have some sort of neuroprotective role in Alzheimer's. However, the protein is highly susceptible to proteolytic cleavage and it's thermodynamically unstable. Additionally, some studies have found that hFGF1 can form amyloid-like aggregates under certain conditions, which renders it biologically inactive. Obviously, this is not desirable. If it does serve some sort of neuroprotective role, we don't want its biological activity to be reduced. So, the relationship between phenylalanine and hFGF1 begins with previous studies that have found that small molecules can actually inhibit the amyloid-like aggregation. So the idea is that phenylalanine might be able to inhibit the aggregation of hFGF1. So to begin this study, I started with a extrinsic fluorescence, a Thioflavin T experiment. Thioflavin T is a cationic benzothiazole dye that binds to aggregates. So after binding to aggregates, there's an elevated relative fluorescence intensity. So I used different concentrations of phenylalanine, and over the course of 30 days we see an increasing relative fluorescence intensity, indicating that aggregation is occurring. Characterization of phenylalanine fibril assembly begins with a study of the effects of SDS, or sodium dodecyl sulfate, on phenylalanine aggregation. So SDS is an amphipathic detergent that mimics the plasma membrane of cells. So basically we just added some SDS, 0.2 millimolar SDS, to the solutions, and we see that the blue graph in the presence of SDS shows elevated fluorescence compared to the orange graph that doesn't have SDS. So this could mean that membrane-like environments are preferential for the aggregation of phenylalanine. And so continuing, we look at the effects of pH on phenylalanine assembly. We do this by changing the pH of the buffer that the samples are made in, which is 10 millimolar phosphate buffer saline. As you can see, the blue graph is pH 2, the orange is pH 7, and the gray is pH 9. The blue graph, pH 7, appears to have a higher fluorescence intensity than the other two conditions, meaning that a positively charged amino acid may be plausibly required for the assembly of phenylalanine. So this could mean that electrostatic interactions play some sort of role in self-assembly. Phenylalanine self-assembly was further explored using transmission electron microscopy. The way this study worked is we used 6 millimolar concentration of phenylalanine and allowed it to incubate at 37 degrees Celsius for 15 days. The fibrils that were formed in that time period were isolated and washed and added to a Form-var carbon 300 mesh copper grid. This was used in the transmission electron microscope. These images were obtained at a magnification of 100,000. You can see discrete, well-organized fibrils that appear to be coated in salts from the buffer saline. What this means is that there could be pi-pi interactions that are allowing for the elongation of the fibrils. The only issue with this idea is that pi-pi interactions have large repulsion due to the electrons in the ring, and that could be why there is a salt coating that is stabilizing those negative repulsions. So from this, we can say that cation-pi interactions may have some sort of role in the self-assembly of these fibrils. We also used transmission electron microscopy in the presence of SDS. And the way this worked is we just added the SDS at the beginning of the incubation period and observed the results. So as you can see in these images that were obtained at 50,000 magnification, we see that there are these black spots right here which represent, or I guess indicate, the presence of SDS micelles that have a hydrophobic interior and hydrophilic exterior. Interestingly, the fibrils elongate out of the micelles, so it could be that the micelles are a nucleation site and that hydrophobic core is preferential for initiation of phenylalanine self-assembly. So hydrophobic interactions may have some sort of role in the nucleation of phenylalanine aggregation. So now to talk a little bit about the effects of phenylalanine self-assemblies on hFGF1. First, we had to purify the protein. The way we did this is we overexpressed hFGF1 in E. coli cells and then purified using affinity chromatography column. This was validated using gel filtration, as can be seen here. In lane 7, we have a pure hFGF1 marker. And then you can see in lane 6, which is the 1500 millimolar fraction of NaCl, we see a band that indicates that hFGF1 eluted out with that fraction. This protein was concentrated and used for this experiment, the seeding kinetics. The way this worked is we took 6 millimolar concentration of phenylalanine and allowed it to incubate for the 15 days at 37 degrees Celsius so that we could obtain those fibrils. The fibrils were then isolated and washed and added to a sample containing hFGF1. We also had a separate sample that had the FGF1 by itself and a separate sample that had the phenylalanine fibrils by themselves. We observed the extrinsic fluorescence of Thioflavin T over the course of 24 hours, and we see that this orange right here is the hFGF1 by itself, the gray is the sample with the seeding aliquot, and then the blue is the phenylalanine fibrils by themselves. We see that the fluorescence intensity of hFGF1 is higher than that of the sample containing the seeding aliquot, indicating that the phenylalanine fibrils reduce the fluorescence intensity and the aggregation of hFGF1. So it could mean that phenylalanine self-assemblies have some sort of an inhibitory effect on FGF1 amyloid-like assembly. So to conclude, phenylalanine forms self-assembled structures in vitro, as is evidenced by the fluorescence kinetics experiments and the transmission electron microscopy experiments. We also see that SDS appears to increase the aggregation propensity of phenylalanine, and that electrostatic and hydrophobic interactions may play some sort of role in the nucleation and elongation of phenylalanine self-assemblies. And then finally, phenylalanine may have some sort of inhibitory effect on hFGF1 aggregation, and this could be a possible technique for modulating the potential neuroprotective behavior of hFGF1. As far as future work goes, we plan on performing some experiments using ANS, which is a dye that binds to solvent-exposed hydrophobic regions. What this will do is allow us to further explore the idea that hydrophobic interactions play some sort of role in phenylalanine self-assembly. And then we would also like to use circular dichroism spectroscopy to explore the idea of secondary structural-like characteristics of these assemblies of phenylalanine. I would like to acknowledge the NIH as well as the U.S. Department of Energy for helping fund this project, as well as the University of Arkansas Honors College for funding my research as well. Thank you.